My name is Claire Bomkamp. I'm a senior scientist with the Good Food Institute. So just a quick background for those of you who are new to GFI. We're a nonprofit who works to advance the science of alternative proteins. And so we do everything from working with scientists and researchers to working with policymakers, and then also working with the for-profit food companies to make sure that these things are making it out into the world and being commercialized in a way that builds a better food system. So just a couple of quick housekeeping items. We are recording this session, and we'll send you the link if you're registered afterwards if you want to go back to anything. If you see at the bottom of the screen, there should be a chat box. You can use that to network, introduce yourself, get to know the other participants, sort of however you like to use that. And then we also have a Q&A. So if you have questions for our speakers, you can use that as a way of making sure things are a little bit organized. That just makes it easier for us to keep track of what's been asked and answered. And so my colleague Jen Lemmy, who manages the Sustainable Seafood Initiative, will be monitoring the chat and Q&A. So we'll have about 20 minutes per speaker, and then a Q&A session after that. So any of your questions that you've put in the chat throughout the talks, we'll go to that. We do have a networking session immediately following this. So it'll be 5 minutes after whatever the next hour is where you are. So you can register for that if you're interested. We also have a RFP out right now that the deadline is November 1st. So we're going to be funding some projects on cell line development for cultivated meat from poultry and seafood, as well as animal-like fats from plant-based and fermentation-derived systems. And if you'd like to keep up to date with other swim events or other seafood shenanigans we have going on at SSI, we have a newsletter. It's quarterly. I promise we don't spam you. So that's the link that I just put in the chat there. And so with that, I'd like to introduce our two panelists. So Dr. Yi Ling is a developmental and stem cell biologist by training and is now the CTO and co-founder of Shiok Meats, which is a Singapore-based company and the first company in the world working on cultivated crustaceans. And they're doing some very cool things there. And then we're going to hear from Professor Andreas Lopata from James Cook University, Singapore. Dr. Lopata is a world leader on seafood allergies and just a really interesting person to chat with. So I think he'll have a lot of really cool things to share. And we're so lucky to have both of these amazing scientists contributing their talents to the growth of this field, making cultivated crustaceans happen, and also making sure that we're doing so in a way that's safe for consumers. And also, of course, very lucky to have them speaking to us tonight. And so with that, I will stop talking and turn it over to Yi.
Great, let me share my screen. Thank you, Claire, and thank you, everyone. I'm really excited to be here and be part of this session that is dedicated to crustaceans. Can you see my screen okay?
Yes.
Great, thank you. Yes, so today I'll share with you guys a little bit about what we do at Shiok Meats and some of the challenges we face on working with crustaceans that some of the colleagues here might also have experience and would love to learn from you guys as well. So I'm Yi, I'm the CTO co-founder of Shiok Meats. So why are we all doing this? Just a brief overview, since most of us have already been plugged into this cultivated meats scene, is that we know that the population is continuously growing, and how do we make sure we can feed everyone, but at the same time not continuously damage the oceans and the earth that we are so limited of. So this is why a big motivation of what we do. And a little bit of overview of the landscape. This is something that the Kind Herb Tech does very often, looking into who are the players in the cultivated scene. And seafood is something that is slowly growing, and we're one of them. And for us, What is unique about what we do at Shiok Meats is that we focus on crustaceans, which includes shrimp, crab, lobster, and crayfish. And our main focus is also the Asia Pacific market. Oops. Sorry, the slides were jumping. And why did we start off with shrimp and also with crustaceans? Is because other than wanting to focus on Asia, where we eat a lot of seafood, and shrimp is one of the most common seafood that we eat, if you look at the dishes that we have, is that there are a lot of issues with shrimp and crustacean production. And a lot of studies that look at the products that we find of shrimp in restaurants and in supermarkets, 30% of them were not what they say they are. It could be a different species, it could be from a different country, and consumers like you and I, when we go to, or lay consumers, when we go to the supermarket or when we eat in a restaurant, it's very difficult for us to tell exactly what species it is just from eye. And then with farming of shrimp and crustaceans, whenever intensive farming is involved, a lot of harmful chemicals like antibiotics, pesticides might be used. And these end up in our oceans and also in the seafood that we eat as well. And with fishing, some of the issues there is that there's a lot of bycatch because the nets they use are very fine. And so for every pound of wild shrimp that it's caught, there's 20 pounds of bycatch that is sacrificed because they're taken out of the ocean while the shrimps are being taken out from the nets. Oops, I'm sorry, guys. I'm not sure why it keeps shifting backwards. Yep. And then in some areas, slave laborers are used to remove the shells for the deshelled shrimp that we have. And then it's very unsustainable to keep going back to the ocean because that takes a lot of time for the crustaceans to grow, and we're not giving enough time for it to replenish at the rate we're eating. And finally, a lot of the wild seafood, including shrimp, digest the microplastics and heavy metals, the pollutions and pollutants found in the ocean. And on the other side, other than the environmental motivation, we're also motivated by the market share of crustaceans. If we look at the market, it's growing continuously and projected by 2025 to grow to 150 billion US dollars, which is a huge amount. Asia, which is our primary market focus, is also a huge place where production is high there, and there's also a lot of interest for the consumption as well. And US and Europe are also major markets. And for processed crustaceans products, Asia and US are also, and Spain are some of the top importers and exporters, which also is one of the reasons why we focus a lot of efforts in the Asia Pacific region. Shrimp, and in particular, has very good nutrition. It has very low calories, and most of this comes from protein. So that's really great in terms of a food source. And Shiok Meats, we are founded in 2018, almost 3 years ago. And when we started, we were the first cell-based cultivated company in Southeast Asia, and also the first cultivated crustacean company globally. We're on a mission to bring sustainable, healthy, cruelty-free, and delicious seafood and meat to the world by harvesting it from cells instead of animals. And if you're not familiar, Shiok in Singapore and Malay slang means delicious or fantastic. Shiok Meats was founded by myself and Sandhya. We both are stem cell biologists with over 20 years of combined experience. Shiok Meats is my first startup, and very happy to have ended up in the cultivated seafood area. Now we've grown our team to over 30 people, of scientists, engineers, food technologists, business professionals from all over the world. And we're constantly looking to grow our team. So if you have any interest in the crustacean area and want to get into the industry, definitely contact me or look up Shiok Meats on our website or LinkedIn. We might have positions that are interesting to you. So why are we doing this? I mean, using cultivated meats as an alternative and additional production process makes us independent. And for countries like Singapore, where we have very limited resources, this gives us the opportunity to produce seafood and meats without relying on natural resources. There's a faster production time. From our process, the production takes about 4 to 6 weeks, whereas to grow a full shrimp, it takes about 6 months, and for lobster and crabs, it takes years. It's more sustainable to the environment. We use less water, land, and energy. We don't need to use harmful chemicals, making the product healthy, and we don't have issues with labor and animal welfare. We're very happy that we started off with just an idea. And when we first started, we had to go to the St. John's Island, which is offshore in Singapore, bring all our little bags of consumables and our animals to the offshore lab, because that was the only space we could rent to do our first experiments. And then we were in A-STAR, the Singapore Bioimaging Consortium, where we were able to share labs with the scientists there. And now we've moved into the first food incubator in Singapore called Innovate 360, where we have several labs and offices, and we are opening our mini production plant soon in the next month. A little bit about the technology. We have patented the technology specific to crustaceans, how we can isolate and make food from the cells we isolate. And what we do is that we isolate muscle stem cells from the live animal, in particular the muscle stem cells, and then provide it with the right nutrients, growing it in larger and larger vessels into bioreactors and providing it with the specific nutrients it needs. And once we grow sufficient mass or numbers of cells, then we differentiate it back into muscle fibers, and then we can collect them and use them to make various dishes. And we have been very fortunate to have been able to work on several different crustaceans, starting off with shrimp in 2019 with our shrimp dumplings, which were quite popular. And then moving on to the lobster, where we ventured into lobster gazpacho and lobster terrine. And just a few months ago, we showcased our crab, where we did the Singapore famous chili crab, and then some crab cakes, and then we also showcased shrimp, tom yum soup, and lobster chips. So all of this, we were very excited to be able to showcase that there were very distinct taste and flavor, just as what we expect from when we eat the animals itself as well. And so for our company timeline, we've been working a lot on the research side, and we're still in the R&D phase. This year, soon we'll be launching our mini production plant, getting us to mid-production scale, and will get us started on our regulations process. And next year, we'll be building our first pilot production plant and working on the approval in Singapore. And hopefully by the start of 2023, we'll be in 1 to 2 high-end restaurants in Singapore. Singapore has been a great place to start. When we started, Singapore wasn't really looking in, alternative proteins as much, but now it's known all over the world. We were the first to approve the Just Chicken for sale, and right now still the only ones in the world approved. There's been a lot of different alternative protein companies starting in Singapore, and Singapore has been trying to attract a lot of the alternative protein companies to come as well. And Singapore consumers have very good positive interest in cell-based or cultivated seafood. As we've done our own study last year, we found that over 78% of Singapore consumers were excited to try cell-based seafood. And in particular, their interest comes from environmental concerns. This year, we did the study in Hong Kong with over 1,200 participants over a large age group. And there's even higher interest in Hong Kong residents to try cultivated meat and seafood of 95%. And that was mainly motivated by food safety. So that's really exciting for us as well, being interested in the APAC market. And we will be launching first in Singapore, but we would also like to establish manufacturing plants in various parts of Asia Pacific, including Australia, Thailand, India, Malaysia, Korea, Japan. And outside of Asia Pacific, we're looking to work with other companies either to license our technology or set up joint ventures and collaborations. We're very into collaborations with other companies and research groups. We've been working with Intericulture to look at more cost-effective nutrient source for cell-based shrimp. And then we've also signed an MOU with the Singapore Polytechnic, which focuses a lot on food innovation. And, as I've mentioned, if any of you are keen to work with us, we're happy to speak further, as there are much to be explored in the crustaceans biology area. We've also acquired recently Gaia Foods, which is Singapore's first structured red meat company. We are keen to work with other companies, but our main focus at Shiok Meats is in crustaceans. Working with other cell-based and cultivated companies gives us the area to expand into other meat types, and also, with their interest in structured meats, we hope that will help advance our work in structured crustaceans as well. We're very fortunate to have raised funding up to date of $30 million, including grants in both our seed and Series A in total. And that's the end of my presentation. Thank you.
Thank you so much, Kai. That was fantastic. I'd like to now turn it over to Professor Andreas. Unfortunately, we've been having some technical difficulties, so I'm going to attempt to share slides.
Yeah, thanks very much. Thanks so much, Claire. Yes.
Okay, are you seeing the slides?
I can see it. Perfect.
Okay, just let me know when to.
Yes. Yeah, thanks so much first to Kai, give the really nice background information for my talk, talking a little bit about the other side of crustaceans. Not necessarily the bad side, but something we can't oversee. And this is certain potential safety issues with people who have existing shellfish allergy. So in the next slide, I just want to show where we are working, James Cook University, and we have the main campus, or the mother campus, is in Australia, right in the north of Queensland. And I'm working now at James Cook University in Singapore, in particular the Tropical Futures Institute, which focuses on the production of seafood in an urban and marine setting. And my particular focus is on the food safety side to bring these seafood products onto the market. The next slide, you can see where we are located. Right in the north, if you don't know Australia so well, that's really in a tropical part, almost as hot as Singapore, not quite. But in the next slide, you can see we are quite situated next to the sea, very green. Clearly, seafood production and seafood research is very central to our interests. And on the next slide, you see the campus in Singapore is more kind of urban environment. So next slide, please. Seafood and safety, it's really, of course, of importance for selling seafood and consuming it safely. But when it comes to any type of adverse reaction to seafood, the vast majority is really in regards to exposure to toxins, exposure to parasites, exposure to heavy metals, and actual seafood allergy is a very, very small part of it. The bad side of this is usually people don't lose this type of reaction. I'll show you just now why. The next slide. You can see that we distinguish basically two types of reactions to food in general. The one is food intolerance, you see in the bottom, which does not involve the immune system, and you have to avoid the food. And if people get occasionally reaction to a particular food or seafood ingestion, but not always, food intolerance and certain adverse reactions is probably what you see. And in contrast, food allergy, you have really an abnormal response of the immune system, and this is often lifelong. In the next slide, please. You see here on the right side all of these kind of toxicological reactions to fish and shellfish, mostly ciguatera, scombroid poisoning, but marine biotoxins we find quite a lot caused by algae, and you find this in shellfish, crustaceans, and filter feeders and a lot. And on the left side, you see seafood allergy is really involved with IgE-mediated reactions, basically involving the immune system. The next slide. You see the more kind of complicated way, but I don't want to give you an immunology lecture. But on the left side, you see any type of food. Peanut is very often the same because peanut also is for 80% of people a lifelong condition. If they get it as a childhood or early adults or late adults. And the same, unfortunately, is also with shellfish and fish as a seafood allergy. And at the end, basically, secondary exposure, once somebody is sensitized, causes these allergic symptoms. And that's what we have to avoid and manage. And the allergenic proteins involved in these kind of reactions are part of our analysis and gives us an idea if certain seafoods are more or less allergenic. And we have clearly evidence that there are differences between them. They're not all the same. It's not like we have a peanut or an egg allergy. All eggs and peanuts are 99% identical worldwide. Whereas when it comes to fish and shellfish or any type of seafood, there are literally thousands of different species consumed and farmed, and now also, of course, cell-based produced worldwide. The next slide, please. It's a fast-growing problem, allergy, unfortunately. Half the population of the world will be in a few years' time sensitized to some type of allergy. Of course, not only food, but mostly pollen and mite. And this might also be a problem for some people with shellfish allergy. But it's about worldwide, maybe 500 plus million people have some type of food allergy. And 25% of this food allergy seems to be mainly directed against the shellfish and fish, because, as I said, in adulthood, most people don't lose this type of allergy. And the next slide. And the next slide, please. You see the big one before, please. The most 90% or more of all food allergic reactions are actually targeted to milk, egg, peanut, tree nut, and so on. And as I said, peanut and tree nut allergies are not lost in adulthood, and fish and shellfish behaves basically very much the same. In contrast, milk and egg allergy, it's a childhood disease, and most adults, I would say, will lose this allergy later on. And fish and shellfish, that's what our research is particularly focusing on. And next slide. What's the prevalence? How often does it occur? And plus minus 3%. In countries where you eat a lot of seafood, this number seems to go up. We have done studies in Vietnam. 5-6% of adults and children have some type of seafood allergy. In Singapore, probably a little bit less. What's the problem? This is, of course, is the serious anaphylactic reaction, where people can really have life-threatening reactions, clinical reactions to it. And on the bottom, I'll show you there are two proteins involved. One is called parvalbumin, and the other one is tropomyosin. And because they're really muscle proteins, they are very important, of course, in the production of any seafood, because muscle tissue is what you want to eat. That's what the protein is, and that's also where the texture of these seafoods are. On the next slide, please. You see, when you look at seafood per se, and when you have a seafood platter, this comes what you see. And we know all of these individuals very nicely, but of course the allergens, when it comes from the allergen point of view, are very, very different. On the left side here in blue, you see fish. We have done extensive work with 60, 70 different types of fish species, particularly from the Asian Pacific region, which are very different from European. As Kai already earlier said, it's a very much focus of our research in the Asia Pacific area. And we found there a huge difference between fish. Some of them are highly allergenic and some of them are super low allergenic, which is of course very nice for food production also to help consumers consuming more types of seafood. When it comes to shellfish on the right side, mollusks on the top and crustaceans on the bottom. Now, when it comes to allergy point of view and also phylogenetically, mollusks and crustaceans are completely unrelated to each other. And this reflects also in the presentation or expression of proteins which make up these organisms. When it comes to allergy, unfortunately, clinicians often say, if you have any allergy to any type of shellfish, don't eat any shellfish. Unfortunately, it's not 100% correct because a lot of people can eat certain types of shellfish or the mollusks or crustaceans, but not others. And I'll show you also the reason for this just now. And in the middle, you see also insects coming up at the bottom. Insects and mites are actually quite closely related to crustaceans, but not really at all to mollusks. And this helps probably also this relationship in growing these tissues easier. The problem is when it comes to allergens, some of them might be very close related. The next slide, please. A closer workup of the different types of groups. When you look on the top, the different prawns, or shrimps, they are not all the same. And we did a very detailed proteomic and genomic comparison of 5 different shrimp species and see if the allergen contents and the variability of these allergens actually is different. And when you look under bivalves, we particularly focus also some research on abalone and oyster, because oysters are eaten often raw, whereas most of the other seafoods are eaten cooked. And we also had a look at the allergen distribution between heated and non-heated products. Next slide, please. Yeah, this is, again, I don't want to bore you with chemistry, but this is basically how it all starts. Analyzing allergens from seafood is looking very detailed on the biochemical analysis, separating all of these proteins, and that involves usually a lot of mass spectrometry to identify any protein and identify particularly the ones which are similar or different between different species. So on the next slide, you can see we did something very intensively with the oyster, as I mentioned earlier on, because they are often eaten raw. And on the left side, you can see we had a bioinformatic approach, an allergenomic approach on the left side where you use patients who have some type of seafood allergy. On the right side, proteomics approach where we investigate all of these thousands of different proteins you find in these extracts. And now more recently in the top green, we're using bioinformatic approach, not only to identify these allergens, but also predict allergenicity. And that's work we're doing with A-Star University, with the Bioinformatics Institute, really intensively to be able to predict allergenicity of certain seafood allergens without doing very extensive clinical studies, which can often take years. But on the top right, when you look at the table, you see between raw and heated oyster products, you find in the heated products only 4 allergens left, because many of these allergens are heat sensitive, which is a good thing. In the raw, in contrast, you find 24 allergens. And on the bottom, we have this big red arrow showing to tropomycin. So tropomycin is a protein which you find really mostly in the heated products, because this is a heat-stable protein. And on the bottom, there are a couple of other allergenic proteins, which usually after heating disappear because they are heat sensitive. And then on the top right, the sarcoplasmic calcium binding protein is another protein that comes later on, seems to be also quite heat stable. The next slide, please. So having done all of this investigation over the past years, we identified quite a few proteins which can cause allergies, only in, as I said, in a very small proportion of people, but it's important to identify them and to warn them that they don't consume products which might cause anaphylaxis. But the importance of these allergens varies dramatically. Some of these allergens are only recognized by 10 or 20% of people with shellfish allergy. However, tropomycin seems to be one of the major ones, and 70–80% of patients can react to these tropomycins. So on the next slide, please. You see now when you have an allergy in the top left, you see the shrimp. And the shrimp tropomycin, if you have allergy to this, there is a highly likelihood that you also react to other ingested crustacean species, but less likely to mollusks. And even when you see on the left side, you see DERP10, this is mite, and also the cockroach, which you see here, they have very similar allergens. So some people, and that's maybe 10, 20, 30% of people which are sensitized to crustacean can also react to inhaled mite allergens. And I'll show you a little bit more about this. So this clinical cross-reactivity based on recognition of a particular food allergen is quite unique when it comes to food allergy per se. You hardly have this with peanut and tree nuts to a certain degree. With egg and milk allergy, you don't have this at all. But when it comes to seafood, the biology behind the species diversity is very significant. The next slide, please. Yeah, so we tried to develop a kind of a flow chart, kind of predictive model, and this was a few years ago. Now we have even better tools to make it more intensively, but we looked at just two different allergens, tropomyosin and arginine kinase. Between oyster, you see in the left, the tiger prawn, nice big ones there, and the mites. And when you look at the top right at this diagram, you will see the mollusk tropomycins. They are completely far away from crustaceans. And crustaceans, in contrast, they cluster, crustacean tropomycins cluster to cockroach and mites. So they're close related. So we looked at another allergen in the bottom, arginine kinase. It's the same kind of profile. Clustering of all these mollusk arginine kinases are very different from the ones from crustaceans. And in the next slide, you can see basically the reasoning for this. In a nutshell, we looked at all small parts of these proteins which causes allergies. And on the right side, this large green block, you can see between all the different crustacean species, these small protein sequences are very identical, meaning cross-reactivity is very high. So if somebody is sensitized to one shrimp, it's very likely also this tropomyosin of another shrimp will also cross-react. When you look on the left side, with mollusks is very different. There's a huge diversity of these different protein tropomycins between the different mollusks. So, and this also probably is the reason that mollusk and crustacean allergy is not really cross-reactive. On the next slide, please. Yeah, here we investigated now very different crustacean species. You see this on the right side, this diagram where we have prawn whole cooked extract and prawn raw extract. So we compared again raw and heated, not only for oyster, but also here for prawns, then for lobster and crab. And again, on the left side, you see tropomyosin in the black bar, seems to be the major allergen, heat stable between all the different crustaceans, but there are quite a few other proteins also heat stable and seem to be relevant. On the right side, in contrast, when you have a product which is not heat treated, arginine kinase, which is a different class of allergen, seems to be predominant. So we did this with mass spectrometry, and when you look at the left side, we then subsequently did a genomic approach, and that's similar to what Yi earlier mentioned. We had now the transcriptome of 5 different shrimp species and compared them for their general protein content, but also for the allergen content. And as Yi said, very important to get all this information also for growing shrimps in culture, but also from the allergenicity point of view. The next slide. These transcriptome analysis basically then enabled us to compare 5 different prawn species, or we call them prawn, but they're basically shrimps. That's another way of calling them. Vanamai on the top left and on the top right, the black tiger prawn. In total, we found about 40 or 44 different allergens in these species using transcriptomic analysis. And then when you look at the bottom right, the Endeavour prawn, for example, had up to 50 different types of allergens. So knowing these allergens and the distribution really clearly showed us even a shrimp looks like a shrimp. When it comes on a molecular level, as you earlier heard already, they are very different. And this reflects probably also in the growing conditions you have to, for each species, to adjust. And the same you see this in natural caught prawns. They're very, very different. Even they look very similar. And definitely once you cook them, the taste also very similar. So the next slide. So we went in one step further and said, okay, let's focus just on black tiger prawn from various different parts of the world, and then compare wild caught and as well as farmed tiger prawn from different parts of the world, if there would be any chance of variability between the allergens in between the species. So we had shrimps from farmed in the wild from India, from China, Indonesia, and various from Australia. On to the next slide. You can see now on the left, again, quite complicated, but on the left side, when you look at the bar graph, Queensland F means Queensland farmed prawns, and Queensland W means Queensland wild caught. And there you see already in blue, the bar in the wild caught is much bigger than in the farmed ones, and this blue represents tropomycin, the heat-stable allergen tropomycin. So even there, between farmed and wild from one country, you see big differences. And when you compare this, just looking at the blue, the tropomycin between the different countries, wild and farmed, again, huge differences in the contents of these tropomycin protein. In particular, India, farmed on the very right, seems to be very different. What is the reason for this? I mean, of course, we don't know directly, but it must be a reflection of environmental conditions. If it's temperature, salinity, or other environmental conditions will affect, of course, the expression of different proteins. And on the right side, we compared farmed versus wild. There seem to be, as you said, particular differences between myosin light chain, tropomycin, sarcoplasmic binding protein, but we found also a whole range of other proteins. And when we look between the countries, again, there are 3 or 4 major allergens always popping up there of importance, but there are definitely huge differences when we look at the origin of these species. The next slide. We went one further. On the left side, hemocyanin is one of the known allergens, which maybe 20% of shellfish allergic people are reacting to. But we looked now at the different isoforms or subunits of these different hemocyanin proteins. And there also, when you look at the very left side here, against Queensland farms and Queensland wild, in pink, there are different distribution of isoforms making up the hemocyanin protein. Again, a reflection of course, environmental condition, because hemocyanin, we know, it's also part of the similar our hemoglobin of transport of oxygen and other molecules in the hemolymph of shrimps. And on the right side, sarcoplasmic calcium binding protein, part of the muscle contraction. Also there, the known different subunits for these particular proteins we know differs between the different origin. So thanks very much, and look in the next slide. So combining these, the knowledge we have now about allergens in seafood or the particular crustaceans, and we heard from Yi, when it comes to allergens, we in a wild and aquaculture is a lot of under-investigated knowledge we have. We have a lot of knowledge gained over the last 20 years, but because the species and the growing conditions changes the whole time, if it's now in a wild caught or urban setting of aquaculture, it will probably change the composition and profile of allergens. And with alternative seafood, we don't know yet, but of course, it opens an opportunity for perhaps controlling allergen contents or distribution in various alternative seafood species. So in the next slide. So just a summary of a couple of things. Sorry, on the left side, things are middle up a little bit, but basically we talk about seafood allergy that it affects around 3% of adults and children. There is also the chance of cross-reactivity because some of these allergens are related to mites. So in population where a high mite allergy is prevalent, like in many Asian Pacific countries, mite allergy is very common because of humidity. There is often this oral allergy syndrome, which I didn't really go in detail, but there is cross-reactivity on the allergenic part. There is no cure for food allergy, in particular for seafood. Maybe some are working now on peanut allergy, which might be coming soon out, immunotherapy, but definitely not for seafood. And ultimately, we still have to fill up a lot of knowledge gaps in the production of alternative seafood meat. And this gives us also, of course, unique opportunities to do better seafood for the future. And the next slide, please. Yeah, this is basically our key capabilities at James Cook University in the Tropical Futures Institute, working on different parts on seafood. On the left side, we worked a lot on prevalence and diagnostic in the medical field to identify the allergens. Now we're working in the food labeling space with government authorities to help them to produce good detection test kits for food products. Quantifying and detecting allergens is very crucial for any export of food products into European or North American markets. And the allergenicity prediction we developed with Singapore A-Star University, and as well as the FRESH organization within A-Star. We're trying to develop methods to predict allergenicity before we have to go into clinical stuff and more detailed studies. Yeah, I think this is the last slide. Thanks very much. And this is my team. Of course, nothing can be done by one person, but I have really a fantastic team over the last 4 years of excellent scientists, really dedicated to food allergy, in particular seafood allergy. And quite a few of ex-PhD students are now doctors and working in Indonesia and Vietnam and various parts of the world. And with this, thanks very much. And I hope there is raised some interest with you, having some questions to follow up. Thanks very much.
Thanks so much, Professor Andreas. That was a great talk and a great intro to sort of the immune system being always more complicated than you think it is.
It is.
So we have just a few minutes for Q&A before we move to the networking session. So first question from Louise, I think it's for Kai. And so he asks, you have already closed the cycle for production of cell aquaculture crustaceans. And can you talk a little bit more about the bottlenecks that we can work on to improve the process?
Great. Thank you for the question. I think we are working to close the cycle. I wouldn't say that we are there yet since we haven't gone to commercial scale. So that's one of the challenge, excuse me, to get to scale is something that is not trivial. Even though it's possible at R&D, how do you get to it in an efficient manner and a cost-friendly manner? Because at the end of the day, this is food. We're not going to spend thousands of dollars for each meal. So that's some of the bottlenecks. And I would say one of the key challenges in research that would be helpful to improving or getting us into a commercial scale would be, for the cost, a lot of it is what we feed the cells, the nutrients, and the culture media we use. And that's not so understood because most of our work has been focused on the mammal side of things. So on seafood and crustaceans, that's not as understood. So that's one. Then the other part would be, for a lot of consumers, we want to see structured products. So how do you get from just mince, which I guess in Asia, that's very popular to make different shrimp balls, dumplings, and all of that. But we all want to see some kind of C-shaped shrimp or a crab claw, a lobster claw. And those are, more difficult challenges that I think there are technologies out there, but also the question of, if we use 3D printing or hydrogels or scaffolds, are these scalable? Are these cost-friendly if we incorporate it into the process? So here are some of the bottlenecks that I think are key to the crustaceans side.
Great, thank you. We have a question from Kevin that I guess is primarily for Professor Andreas. And so she says, it would be very interesting if you could test the cellular-based crustacean material that Shiok has developed. How much material do you actually need to do the allergen testing?
Yeah, Kai, good question. So, you don't need much material. Also, basically what you eat, if you have a prawn dumpling, what's in there, it's enough to do a full analysis using biochemical methods, which we have, and you should get a good idea of what's present there. But of course, the first question is, what do you test? Is this a product which you have in tissue culture or after heat processing and what you get on your plate? Because there will be differences. And how is it heat treated? And if you have a prawn-shaped product, it might be different heat treated and produced than a minced up dumpling, which gets into hot water for 5 minutes or something. So that's also this food processing part is something we need also to take into account, not just the product in a petri dish. And then, of course, the other products around it is also the scaffold. I mean, that's also discussion. Is the scaffold completely non-allergenic, or could be some of these plant-based scaffolds now be a new source of plant allergens in a seafood product? So these are discussions which are already up there, not only for seafood, for other types of meat. It's the same questions coming up with chicken and beef and so on, how we do this, and analyzing it's the best way to do it. Also having consumer confidence that it has been done, we analyzed it, and we found nothing. That's the best outcome. But now having this huge databases, we can look at literally millions of different proteins in a product and can predict if some of them could be an allergen or not. Yeah, I hope this helps. Yeah.
Yeah, sounds like there's sort of a lot of nuances, and it's not just a matter of you need the material to do one test, but you need to test and test and test.
Yeah.
Another question from Rohit: how would we avoid clean meat allergic reaction if it's a basic part of the protein machinery in crustacean seafood? So I think that's
yeah, look, I mean, that's a big advantage, as I said, cell-based meat is that you can alter growing conditions. And while we're in a moment in the stage worldwide of identifying what kind of allergens are found in different species, you could identify certain species which are low in certain allergens. You can modulate even the species, not even the growing condition. But species plus growing condition together gives you an even more powerful way of looking at these allergens. But in a nutshell is, if you have, in a nutshell or in a shellfish thing, if you have an allergy to shellfish, you cannot avoid it. You know, you can treat it with immunotherapy, or you choose a product which has no shellfish allergens in it. So if it has the same type of allergens as any normal shrimp, there is no concern for 99% of the consumer. But for the 1% which do have this allergy, it could be a problem. So it comes again down to food labeling. So the authorities and governments, they will at some stage when it comes on the plate to say, okay, how do we deal with this? Do we test it? Do we label it? And most likely, yes. I mean, it's not a non-allergenic per se, and nothing really is at the end of the day. It's not just shellfish. Any plant allergen can be as deadly or nutritious. Yeah, that's in a nutshell. Maybe that explains this.
Great. Yeah, and I agree that the labeling is going to be a huge thing that the industry will need to figure out. So we have a question for Kai from Timo. Very exciting and promising. Thanks for the fantastic overview. And they asked, how do you choose the crustacean species for Shiok's R&D effort?
Yeah, for us, I mean, one of the most practical criteria would be the availability. So we have to be able to get live animals. We cannot go to, frozen or refrigerated ones to get cell lines. They're just not good, and we won't be able to propagate them. So that's one consideration. The other one is consumer familiarity. So species that we already commonly eat, like the tiger prawns, the Pacific white, or like mud crab, all of these that we're familiar with. Although I would have to say, from a scientific or a food science point of view, there is no study yet, or we're not sure if different species might really taste different because there are species differences. But I think Andreas said that, for some of the wild caught shrimps, they don't actually taste different after you cook it. So it might be only very expert consumer tasting panels who might be able to differentiate it.
Great answer. Okay. We've got one final question for Andreas. Do B-cell epitopes of the same allergen, for example, tropomyosin, differ across shellfish species?
So sorry, what was the end of it? B-cell epitopes?
Do B-cell epitopes of the same allergen differ across shellfish species?
Yes. There are. We looked at 90 different B-cell epitopes in tropomyosin. It's about 300 amino acid long. We looked at 90, all published data, so not even generated by us. We looked at the literature. And of course, there are differences. And some of these epitopes you find also in mollusks, and you find in lots of other crustaceans. So if you have antibodies against this particular epitope, you can predict already you will cross-react to all the other shellfish and mollusks who have the same epitope. But mostly people have antibodies to epitopes which are crustacean specific and don't cross-react. So yes, there are, I would say, at least 6 or 8 epitopes which are very well characterized and really found in most species, but there are more than we know. Yeah.
Great, thank you. All right, well, with that, I think we're about at time. Thank you so much, both of you, for taking the time to share your expertise and your knowledge. And I hope everyone in the audience learned something and enjoyed yourself. And please do join us for the networking session if that's of interest. Take care, everyone.
Thanks so much. Bye-bye.
Thank you. Bye.
Bye.